Exploring Interdisciplinary Connections, BRCA Gene and AP Biology Unit Connections. Hi, my name is Carter Wilson, and I am currently a senior at Cathedral Catholic High School, and next year I plan to attend the University of California, Davis, to major in biochemistry, focusing more on the molecular biology that goes into the viticulture and enology study. This case is particularly impactful to me because my mother has mutations in her BCRA gene that she is aware of and that are indicative of hereditary issues within her family because of breast cancer. Because she was adopted, she did opt to get genetically tested, but this lab has prompted me to explore the bioethical concerns that coincide with that sort of genetic testing.
Hi, I'm Mason Murphy. I'm a junior at Cchs, and I'm looking to attend Westbourne UCC in Ireland to pursue medical education. This case is incredibly important to me, as many of my family and friends have been affected by breast cancer. As breast cancer is becoming more common, it is important to find more ways to prevent and combat breast cancer.
Unit 1, the chemistry of life. So an essential thing to notice here is that organisms utilize 4 macromolecules in order to live. These are carbohydrates, lipids, proteins, and nucleic acids. Key thing to notice here is that they all include carbon, hydrogen, and oxygen. Nucleic acids are the monomer that make up DNA. By manipulating these building blocks of DNA, these monomers, we can get measurable genetic information such as the frequency and number of nitrogenous bases can tell us a little bit about heredity and relatedness. Nucleotide monomers, as in the phosphodiester bonds that hold together a strand, the sugar phosphate backbone. By noticing the certain schedule and patterning of them, you can get a little insight into what protein or structure that genetic information will eventually code for. And then a little bit about gel electrophoresis, since we're now shifting towards looking how the chemistry of life fits into our lab. It's a tool used to compare fragments of DNA. So fragments of similar size are indicative of shared genetic characteristics. And in this lab, we're comparing individuals' BRCA alleles to BRCA allele associated with breast cancer. Specifically, if the allele is shortened, it's likely that the mutation occurred because that frame shifting mutation caused the reading frame to shift and shorten the allele shorten the fragment of DNA, versus if the fragment stays to the back, it's larger, which means it's not likely that a mutation caused the reading frame to shift. So this is all very interesting stuff that we can notice here that applies from the chemistry of life into our new lab here.
Unit 2, cell structure and function. Cells are made up of several different components: plasma membrane, mitochondrion, nucleus, and more. Cells which become fully mature do not grow. Cells which are affected by cancer do not fully mature. The cells will simply continue growing and replicating. Mutated Brca genes result in non-mature cells proliferating. Unit 3, cellular energetics. Enzymes in the cells regulate cellular replication. These enzymes can be inhibited or activated. Enzymes have active sites which are determined by the structure of the enzyme. Functions of the enzyme work through the active sites. The structure of the enzymes can be changed. A change in structure changes the active site, and the enzyme won't function properly. The Brca 1 and 2 enzymes are tumor suppressors. Without proper function, cancerous cells won't be stopped from replicating. Unit 4, cell cycle and cell communication. Cell cycle regulates cell replication. Interphase is the growth and replication of cell DNA. There are several checkpoints throughout the cell cycle. These checkpoints prevent incorrect and excess replication. Failure to pass these checkpoints results in cell death. Mutations can cause irregulation of checkpoints. Irregulation of checkpoints causes excess replication and then cancer. Breast cancer cells are a direct result of irregularity of the cell cycle. Unit 5, heredity. Inheritance of the mutations only occurs through gamete cells. Gamete cells are reproductive cells like sperm or egg. Mutations in the stomatic cells will not be passed down. Inheritance of genes can be autosomal, X-linked, or mitochondrial. Inheritance of the Brca genes are autosomal dominant. Brca1 is on the chromosome number 17, and Brca2 is on chromosome number 13. Those who are heterozygous for Brca genes are affected. Unit 6, Gene Expression and Regulation. Gene expression is regulated by feedback systems. These feedback systems rely on proteins and messengers such as ligands. These proteins can be manipulated. The proteins can become denatured, and ligands can become permanently bound to proteins. Genes can be unable to be transcribed. The Brca1 and 2 gene suppressors are tumor suppressors. Inhibition of these genes results in excess replication.
So Unit 7 is natural selection, specifically evolution. So evolution is changes in allele frequencies that happen over time. And it's actually any change in allele frequencies, can even be over one generation. As long as there's a change in allele frequencies, that's considered evolution. This specifically includes mutations, which are the main driver of evolution. But remember that mutations occur randomly. You can't really predict mutations. But non-advantageous mutations are usually selected against via the death of an organism if the mutation is that non-advantageous. Mutations in certain regulatory genes can cause cancer. So breast cancer can be caused when the BRCA1 or 2 tumor suppressor genes are mutated. So after they've been mutated, that frameshift mutation results in the regulatory protein that is BRCA1 or 2 tumor suppressor can't properly regulate the cell cycle, therefore goes out of control, cancer occurs, failure to regulate. Are humans purveying natural selection? So if humans can determine who will pass down non-advantageous or cancer-causing genes, will they then ostracize them from society? This unit really makes us wonder about that and ask that question specifically with regards to the bioethics of this, because if it's government mandated to get a test, what will happen to your social credit and your livelihood if you're found at a young age to have breast cancer, heightened predisposition, or other sorts of cancers? Will you be ostracized and people not want to be around you just because of those features? So it's a certain thing you want to think about for sure. So lastly, we have Unit 8, which is about ecology. And this is looking at biointeractions through a very macro perspective. So I went and focused on what major things changed the world and changed human livelihood. And I thought immediately the Industrial Revolution. Giant increase in human population size, waste products began being excreted on such great scales into the environment, greater than just random seismic activity, increased chemical exposure from obviously the industrial aspect of the Industrial Revolution. And the Industrial Revolution placed humans at the top of the trophic pyramid ultimately, because even though we don't necessarily consume alpha predators like hawks and bears and lions, our actions championed by the Industrial Revolution ultimately knock them down a peg, as our footprint on the environment harms them and limits them. Another thing to think about is estrogen hormones in drinking water. So this was mentioned in class by Mrs. Lapaka as one possible link to why breast cancer has increased over the years, is that more young women are on birth control, which is leaving excess hormones in the water supply, compounding hormones in people's bodies and increasing chances of breast cancer in men and women. But many scientific studies argue that the impact of this increased estrogenicity in drinking water is negligible to humans, But other studies have shown that organisms that live in water runoff in urban areas experience high cancer and infertility rates as a result of this chemical and hormonal pollution. So while not humans yet, many scientists argue, other animals are experiencing these cancers as a result of increased hormones in the water. And how many people can planet Earth support? If more diseases and competition is eliminated, what regulates human life? Our population and life expectancies have skyrocketed since the Industrial Revolution, and life expectancies have gone way up, and it's a question of how far is too far, how much can the Earth really support? So my bioethics slide is about the psychological concerns regarding genetic testing. I took Mr. Caro's college psychology class my junior year, and in that class we did a big research project, and mine specifically focused on psychological health and the impacts of that on physical traumatic injuries in the hospital setting. And ultimately all my research concluded that positive mental health outlooks were heavily correlated to positive physical rehabilitation on a numerous of traumatic injuries. So in conversations with my dad as well, who has a PharmD and has a lot of time in the hospital, he agrees with me that psychological livelihood is very important when addressing these sort of traumatic genetic disorders like cancer. But there's two main standpoints, one being that individuals with psychological dysfunctions such as anxiety or depression could react poorly to the knowledge that they have a genetic disorder. This could amplify their anxiety or even cause suicidal ideation as a result of finding out that they have a predisposition to cancer. But the other argument is that people should know that they have a genetic issue so that they can bend their mind around that and confront the issue head on with their mindset and have a better outlook. And that is what my research supports, that with a better mental health outlook, they'd be better equipped to handle the disorder with a positive outlook, often associated with better patient outcomes.
Bioethics slide number 2. Brca testing can lead to genetic discrimination in finding partners. Side 1 says testing can help prevent the spread of breast cancer. Carrier can become aware and choose not to have kids. Side 2 says those who have tested positive for mutations may struggle to find partners, in fear of spreading Brca mutations.
Thank you for listening into our presentation.